So I want you to just imagine the steam rising off a hot paper-wrapped fast food burger.
Okay.
Like, the exact kind I mean. And in the next 5 minutes, as that translucent layer of grease hits the inside of the wrapper, this microscopic chemical transfer gets initiated.
Right. The migration.
Exactly. A synthetic molecule, which was literally engineered in a laboratory to be virtually indestructible, starts migrating from the packaging right into the lipid structure of your food.
It's a very fast process. Yeah.
And within an hour, it deposits into your bloodstream. And because of the specific geometry of its carbon-fluorine bonds, it's just going to continue to circulate through your heart and your liver and your kidneys until like the year 2034.
Which is a staggering timeline to think about.
It really is. I mean, we usually think of eating as this really simple mechanical transaction. You feel hungry, you consume food, the hunger dissipates, and you just move on with your day.
That's the baseline assumption, yeah.
But today, for this deep dive, we are looking at a stack of data that completely shatters the illusion of that closed loop. We have this compelling and honestly deeply unsettling collection of medical trials, occupational health data, and regulatory reports.
Some really massive recent publications in there.
Yeah, including a 2025 GAO report and a 2026 toxicological profile from the ATSDR. And what they reveal is not that we're just, making poor dietary choices. It's that the fundamental biological loop of eating has been structurally hijacked.
And to understand the mechanics of that hijacking, we really have to establish exactly what we are and, frankly, what we are not evaluating today.
Right.
Because when people hear we are analyzing food and processing, there's this immediate expectation of a dietary lecture.
Oh, absolutely. They brace for a conversation about caloric deficits or macros or, the moral failings of not eating enough leafy greens.
Exactly. And we are completely discarding all of that.
Yeah, that's not what this is.
Not at all. This is strictly an investigation into structural biology and toxicology. We're examining how the highly engineered architecture of ultra-processed foods, or UPFs, operates in tandem with synthetic forever chemicals.
They work together.
Right. These compounds are not passive contaminants. They are active agents that are rewriting our physiological baselines, and they completely bypass our conscious choice.
Which means we really need to start with the physical evidence, the biomonitoring data, because we aren't talking about abstract risk models here, right? We're talking about what is physically measurable in the serum of the population right now.
The bloodstream inventory.
Yes, the invisible ledger. And the 2025 study out of UNC Chapel Hill is really the anchor for this. They utilized NAAASE Day, the National Health and Nutrition Examination Survey.
Which is essential.
Right, because it's essentially the gold standard for tracking population health. It doesn't just rely on asking people what they remember eating last week. It actually takes their blood.
Yeah, NATs utilizes high-resolution mass spectrometry to analyze the blood serum of thousands of participants. And then they cross-reference those chemical profiles with extensive, verified dietary tracking.
And the UNC researchers found a very clear signal.
Undeniable, really. They proved a direct, dose-dependent link between the consumption of ultra-processed foods and elevated serum concentrations of specific PFAS.
Per and polyfluoroalkyl substances.
Exactly. And specifically, they were tracking compounds designated as PFHXS and MFOSA.
And I want to break down why those specific compounds matter because, for anyone listening, a random chemical acronym doesn't carry much weight until you understand its physical architecture.
Right, you have to look at the chemistry.
PFAS are defined by the carbon-fluorine bond. And from a purely chemical engineering standpoint, it is one of the strongest bonds in organic chemistry.
It's incredibly stable.
Yeah, it requires immense energy to create and subsequently immense energy to break, which is brilliant if you're manufacturing aviation fluid or industrial nonstick coatings. But biology doesn't operate at the temperatures required to break that bond.
No, it certainly doesn't. And that is the crux of the toxicological profile released by the ATSDR in 2026.
The Agency for Toxic Substances and Disease Registry.
Right. They didn't just catalog the presence of these chemicals, they mapped their pharmacokinetics, basically how the human body absorbs, distributes, and attempts to excrete them.
And fails to excrete them.
Miserably, yes. Because the human liver and kidneys evolved to metabolize and filter naturally occurring organic compounds. They are entirely unequipped to cleave that synthetic carbon-fluorine bond.
It's like bringing a plastic knife to cut through solid steel.
Exactly. The enzymes that break down naturally occurring toxins just bounce right off these synthetic molecules.
And because the kidneys can't recognize them or dismantle them, they just end up reabsorbing them back into the bloodstream instead of flushing them out through urine.
Which leads to the timeline the ATSDR published.
Right, the half-life data. And I really want you listening right now to let this settle. The biological half-life of these specific major PFAS compounds in the human body is between 3 to 8 years.
That's a massive window.
It is. I mean, the half-life of caffeine is roughly 5 hours. The half-life of ibuprofen is about 2 hours.
Right.
So if you ingest these PFAS compounds today, it takes your body up to 8 years to filter out just half of that specific dose. And that's assuming your exposure drops to absolute zero for the rest of that decade.
Which, as we know, is a statistical impossibility in the modern environment.
Yeah, completely impossible.
Because of that immense half-life and the constant daily re-exposure through food packaging, the ATSDR confirmed that these compounds are now detected in approximately 98% of American serum samples.
98%. Wow.
Yeah. We have basically reached a state of near universal systemic saturation.
But the mechanism of how it actually gets from the packaging into the bloodstream is what really fascinates me, because it relies on the exact properties of the food we actually crave.
The mechanics of migration.
Right. People might assume that chemical contamination happens at, like, some industrial plant where a vat just spills into the food supply, but the migration is happening literally in your hands or in your microwave. Let's talk about the thermodynamics of that transfer.
Well, the entry points are deliberately unremarkable. We are talking about grease-resistant wrappers, microwave popcorn bags, and those molded fiber fast food bowls.
Oh, the ones that look remarkably like natural cardboard but somehow magically hold boiling hot, oily food without dissolving?
Those exact ones, yeah. To achieve that performance, the fibers are heavily impregnated with PFAS. The engineering goal is to create an impenetrable barrier.
Right.
But the conditions required to serve the food, high heat, high fat content, and time, those are the exact thermodynamic drivers of chemical migration.
Because fat is a solvent.
Precisely. Lipids are highly effective solvents for these specific hydrophobic chemicals. So when you place a hot, high-fat food item into one of these treated containers, the thermal energy increases the molecular volatility.
So the fat basically reaches into the microscopic structure of the packaging?
And it extracts the PFAS molecules.
Right.
And the longer the food sits, say during a 20-minute delivery drive, the more extensive the chemical transfer.
I mean, you eat it.
You consume the food. And because the chemicals are now suspended in the dietary fat, they are rapidly absorbed through the intestinal wall directly into systemic circulation.
So we've basically established the ledger. The chemicals enter through the fat, they bind to proteins in the blood, and they circulate for years.
Right.
But they aren't just inertly taking up space. And this is where we move from the mere presence of the chemicals to their biological cost. The ATSDR data outlines this multi-system degradation.
Yes, it's not an acute toxicity.
Right, you don't eat a wrapper and end up in the ER. It's this insidious, slow rebalancing.
It is a continuous, low-level physiological strain. The epidemiological outcomes linked to this chronic exposure show a suppressed immune response.
Meaning what exactly?
Meaning the body's antibody response to vaccines and natural pathogens is measurably dampened. And we also see altered lipid metabolisms, which results in elevated LDL cholesterol.
And the kidneys are taking a hit too.
A heavy, constant burden on the renal system, yeah. As the kidneys just futilely attempt to filter compounds they physically cannot process.
Okay, so if it takes 3 to 8 years to clear just half of these chemicals, are we basically running a biological deficit we can never pay off if we keep eating from these packages?
That's the reality of the mathematical model, yes. We are accumulating debt faster than the biological systems can process the payments.
So if these chemicals are the ledger, the food itself is what is corrupting the accounting software.
That's a good way to frame it.
Because if we are going to understand how this loop is hijacked, we really have to look at the 2024 cell metabolism trial. If these foods are delivering the chemicals, why can't we just decide to eat less of them?
The old willpower argument.
I hear this constantly. Just exercise willpower.
Yeah.
But the clinical data from this trial just obliterates the entire concept of willpower.
It really does. The 2024 Kevin Hall feeding trial, published in Cell Metabolism, is perhaps the most rigorous demonstration of this in modern nutritional science.
The methodology is crazy.
It's what makes it unassailable. This wasn't an observational survey where people just vaguely tried to remember what they ate last week.
Right, like I think I had a salad on Tuesday.
Exactly. It was a randomized, controlled, inpatient feeding trial. They literally confined the participants to a clinical metabolic ward. They controlled, They weighed and they measured every single gram of food that went in,
And the brilliant part of the design to me is that they matched the diets perfectly.
down to the macronutrient.
Yeah.
Hmm.
They had an ultra-processed diet and a minimally processed diet, but they ensured both diets were identical in presented calories, identical in total carbohydrates, identical in sugar, fat, and fiber.
Everything was matched.
So if the whole calories in, calories out model were the only variable, both groups should have eaten the exact same amount.
Exactly. The nutritional profiles on paper were absolutely indistinguishable. The only variable was the architecture of the food itself, how it was physically processed.
And they were allowed to eat as much as they wanted.
Yes, the participants were instructed to eat ad libitum, meaning they could eat as much or as little as they wanted until they felt satisfied.
And the outcome was just staggering.
It was.
The participants on the ultra-processed diet unknowingly consumed roughly 500 additional calories every single day compared to when they were on the minimally processed diet.
500 calories.
That is the equivalent of an entire extra meal just slipping completely past their biological radar.
And what is really critical here is the psychological reporting from those participants.
Because they didn't feel like they were stuffing themselves, right?
Not at all.
Yeah.
The UPF group did not report feeling any hungrier than the control group. They did not feel like they were overeating.
Wow.
Their intake increased by 500 calories, but their conscious recognition of that intake remained totally flat. The biological feedback loop that triggers satiety, that feeling of fullness, was entirely bypassed.
Which naturally brings up the obvious question, how? If the sugar and the fat and the fiber are identical, how does the body get tricked into missing 500 calories?
You have to look at the physical engineering of palatability.
Right, because UPFs are subjected to immense industrial pressures. We're talking processes like extrusion, where raw ingredients are pulverized, heated, and blasted through molds to create uniform shapes.
And that fundamentally destroys the cellular matrix of the food.
Right, so by destroying that cellular matrix, the food is essentially pre-digested.
Precisely. When you eat a whole food, say an apple or a handful of raw almonds, your body has to expend energy and, crucially, time to mechanically break down those cellular walls through chewing.
And then chemically break them down in the gut.
Yes, through enzymatic action. Ultra-processed foods require virtually none of that. The texture is engineered to dissolve rapidly in the mouth.
It totally alters the oral processing rate. You can swallow a highly processed pastry or, a handful of engineered chips in a fraction of the time it takes to chew a piece of lean protein or a complex vegetable.
And that speed is the mechanical lever of the metabolic override.
How so?
Well, the human gastrointestinal tract relies on a cascade of hormonal signals to tell the brain energy has been received, initiates satiety.
Right, hormones like GLP-1 and PYY.
Exactly. And those hormones take time to release and travel to the hypothalamus. If you are consuming food whose cellular matrix has been obliterated, you can ingest 1,000 calories in 5 minutes.
So the mechanical intake just outpaces the biochemical signaling.
Entirely. The brain literally does not have the time to recognize the caloric load and apply the brakes before the plate is completely empty.
So the mechanical structure of the food is basically functioning as a speed trap, bypassing the timing of our hormonal signals.
Yes.
But here is where the sources converge in a truly terrifying way. We aren't just dealing with the mechanical speed of the food.
No, we have the chemical aspect too.
Right, because the 2025 Environment International study demonstrates that the PFAS migrating from the packaging are actively dismantling those exact same satiety signals at the molecular level.
This is the real intersection of the structural engineering and the chemical toxicology.
Yeah.
The Environment International study utilized advanced molecular assays to observe how PFAS compounds interact with human endocrine pathways. And they found profound disruptions in thyroid function and, more importantly for our discussion, leptin signaling.
Okay, let's talk about leptin because it's the master regulator. For you listening, if you want to understand why you feel full, leptin is essentially the biochemical brake pedal.
That's a perfect analogy.
It's a hormone produced by your adipose tissue, your fat cells. When you have consumed enough energy, your fat cells release leptin into your bloodstream.
And it travels to your brain.
Specifically the hypothalamus, right. And it binds to receptors there, essentially delivering the message, okay, we have sufficient energy reserves, you can stop feeling hungry now, and you can burn energy at a normal metabolic rate.
And that locking key mechanism is exactly how mammalian metabolism has maintained equilibrium for literally millions of years.
But the molecular structure of PFAS interferes directly with that pathway.
It does. Because these synthetic chemicals are highly persistent and they just circulate endlessly in the serum, they interact with those same cellular receptors.
The leptin receptor.
Yes. The research indicates they can act as endocrine disruptors by either binding to the receptors, physically blocking the leptin from attaching, or by altering the downstream signaling cascade once the receptor is activated.
But wait, let me push back here just a little. If leptin is just a signal and the brain isn't receiving it because a PFAS molecule is blocking the receptor, doesn't the body just naturally produce more leptin to compensate? Like, shouting louder if the receiver is deaf.
That is exactly what happens, and it leads to a condition called leptin resistance.
Ah, okay.
The fat cells sense that the brain isn't getting the message, so they pump out higher and higher volumes of leptin. You end up with hyperleptinemia, which is excessively high levels of the satiety hormone in the blood.
But the brain still can't hear it.
The brain remains completely blind to it because the signaling pathways are chemically disrupted by the environmental pollutants. So the brain looks at the blocked receptors and incorrectly assumes the body is actually starving.
Oh, wow.
And that drives the individual to consume more calories to prevent starvation.
So if the food is engineered to bypass satiety and the chemicals from the packaging are actively scrambling the hormones that tell us we're full, is the concept of dietary willpower fundamentally broken?
It is biologically irrelevant in this context.
You cannot willpower your way out of a broken biological sensor. The body isn't failing. The inputs have literally been weaponized.
It is a systemic override. You're basically asking conscious discipline to overcome a structurally engineered biological deficit.
Which is impossible.
And to understand the scale of this, we just have to look at the 2025 Lancet meta-analysis. They aggregated massive cohort data on UPF consumption.
And what did they find?
They identified a linear dose-response relationship. For every 10% increase in ultra-processed food consumption, there is a corresponding 15% rise in all-cause mortality risk.
A 15% rise. That is a mathematical certainty.
It is.
And if we accept that these foods and chemicals are hijacking our metabolism, we really have to look at who is most subjected to this engineering because, we don't all experience this food environment equally.
We absolutely do not.
We need to look at the structural reality of labor. And that brings us to the 2025 cross-sectional study of the logistics workforce.
This brings all that biological data we just discussed into the socioeconomic reality.
Right.
The researchers analyzed a massive cohort, over 12,000 transport and warehouse workers.
So these are the demographics that maintain the modern distribution economy.
Exactly. Long-haul truck drivers, overnight warehouse selectors, the people keeping global freight moving 24 hours a day.
And the data they found is pretty bleak.
Very bleak.
Within this specific workforce, higher ultra-processed food intake was associated with a 37% increase in the prevalence of both hypertension and fatty liver disease.
And it goes further than just the metabolic markers.
Right, because the biomonitoring data showed that these workers had significantly elevated PFAS body burdens compared to the general population baseline.
And to understand why this specific population carries such a heavy metabolic and toxicological burden, we have to examine the architecture of their occupational environment.
How their days are structured.
Right. Human behavior is dictated by environmental constraints. If you are operating a commercial vehicle for 11 hours a day, restricted by federal electronic logging devices, your route strictly dictates your access.
You are interacting with the food system entirely at highway interchanges, fuel islands, and industrial parks.
And what is available there?
Right. I always hear people use the analogy of, treating the body like a combustion engine to describe this. Like, you just pour cheap fuel in to keep the truck running.
That's a common metaphor, yeah.
But I feel like that metaphor is almost too passive based on this data. It's more like treating the human body like an industrial battery.
Okay, expand on that.
Well, you are hooking it up to a high voltage, fast charging station every few hours. It charges instantly, right? The sugar and caffeine hit your system. You get the immediate voltage required to stay awake and finish that 14-hour shift.
Fast calories, fast energy.
Exactly. But that fast charging voltage is highly corrosive. It runs incredibly hot. It degrades the internal housing, it fries the internal sensors, and it drastically reduces the overall lifespan of the battery.
Right.
But the system doesn't care if the battery dies in 10 years, as long as it powers the machine today and meets the quarterly delivery metrics.
That is a highly accurate, if sobering, assessment of the occupational dynamic.
It's just the reality of the structure.
These workers are not choosing this diet out of a lack of nutritional education. They are selecting foods that serve a direct occupational utility.
They need it to be fast.
They require shelf stability, extreme convenience, and immediate caloric availability. The environment standardizes their intake around what is fast and durable.
So the system extracts the human labor, facilitated by the ultra-processed fuel.
But the biological cost, the metabolic disruption, the hypertension, the accumulation of PFAS, all of that is entirely externalized onto the worker.
Which naturally brings us to the failure of the safeguard.
The regulatory side.
Yeah, if the mechanism of leptin disruption is understood, if the half-life of PFAS is fully known, and if we have clear data showing a 37% spike in fatty liver disease among logistics workers, why hasn't the FDA intervened?
It's a complex question.
I mean, think about it. The FDA has successfully banned trans fats. They have heavily regulated specific food dyes in the past. They clearly have the power to pull ingredients from the market.
They do have statutory authority, yes.
So why is this specific class of chemicals and these hyper-processed architectures allowed to just dominate the entire supply chain?
To understand that, you have to look at the specific legal mechanism the FDA relies upon to regulate food additives, which is fundamentally different from how it regulates, say, pharmaceuticals.
Okay.
We are dealing with the GRAS loophole. GRAS, which stands for Generally Recognized as Safe. And this stems all the way back to the Food Additives Amendment of 1958.
Wait, 1958, we were regulating 21st century synthetic chemistry with a law signed by Eisenhower.
Yes, we are.
That's insane.
Well, the original intent of GRAS was somewhat pragmatic. It was designed to exempt common, universally understood ingredients like vinegar, salt, and baking soda from needing millions of dollars in clinical trials just to prove they were safe.
Okay, that makes sense. You don't need a 10-year trial for salt.
Right. But over the decades, the food and chemical industries realized this exemption could be weaponized.
How so?
Under the modern application of GRAS, a company can introduce a novel chemical additive or a new synthetic packaging compound into the food supply without any independent pre-market review by the FDA.
Wait, I need to make sure I'm following the exact sequence of events here.
Sure.
So a chemical company engineers a new water-resistant compound. Instead of submitting it to the FDA for a multi-year clinical trial, they just, convene their own panel?
Uh-huh.
How does an industry scientist just declare something safe without government oversight?
They utilize what are essentially secret GRAS panels.
Secret panels?
Basically, yes. The manufacturer hires a group of private scientists, who are often consultants whose primary income is derived from the industry itself, to evaluate the chemical.
Okay.
These scientists review the manufacturer's own internal data and conclude that the chemical is generally recognized as safe. And the manufacturer is not even legally required to notify the FDA that they have made this determination.
Wait, seriously?
Seriously.
Yeah.
They can just start utilizing the chemical in the supply chain immediately.
Wait, if GRAS stands for generally recognized as safe, but the industry is the one doing the recognizing, isn't that a massive conflict of interest?
It is the definition of a conflict of interest.
How is safety functioning as a protection here, rather than just a performance? It sounds like total abdication of regulatory authority.
It is. And the 2025 GAO report extensively documented exactly how catastrophic this loophole has become.
The Government Accountability Office?
Yes. Their report was a scathing indictment of this framework. It highlighted the profound gaps in transparency. The FDA literally does not know how many GRA substances are currently in the food supply.
Because they aren't legally required to be notified of all of them.
Exactly. The GAO explicitly pointed out that even when safety data is submitted voluntarily, the FDA lacks the resources and the independent clinical data to verify it.
So the industry just grades its own homework.
And independent oversight organizations have repeatedly recommended closing this loophole, yet implementation remains perpetually stalled due to industry lobbying.
And when it comes to the specific migration of PFAS from packaging into food, the GAO noted that monitoring remains largely voluntary.
Voluntary monitoring for forever chemicals.
Right. But we know the industry has the data. The thermal dynamics of fat acting as a solvent for chemical migration isn't a new discovery. They know the thresholds.
Oh, they do. This is what toxicologists and policy analysts refer to as the management of visibility.
Management of visibility. I like that phrase.
It's essentially a form of regulatory theater. The agencies do not deny the existence of the risk because denying it outright would invite immediate legal and scientific backlash.
Right, the data is too obvious.
Instead, they manage the public's perception of the risk. They issue public acknowledgments, they call for voluntary phase-outs, they announce that they are funding ongoing, multi-year studies to better understand the pathways.
Oh, I see. They use the scientific process itself as a shield against taking regulatory action. We can't regulate yet, we need more data.
Exactly. But they've been gathering data for decades.
Right.
By constantly shifting the goalposts and keeping the chemicals in a state of pending review, they create a public cadence that feels like responsible action, but is actually deferred action,
It maintains the illusion of safety.
while ensuring the underlying flow of commerce, the manufacturing of the packaging, and the distribution of the food remains entirely uninterrupted.
And this is where the timeline becomes horrifying. Because while the regulators spend a decade negotiating voluntary phase-outs and waiting for another longitudinal study to finish, the chemicals aren't waiting.
No, they are accumulating.
They are. And because of the 3 to 8 year half-life, this regulatory theater isn't just a bureaucratic failure, it is a biological sentence.
A generational one, actually.
Which brings us to the most profound data in our stack of sources today. We have to look at what happens when this exposure outlasts us. We have to look at the epigenetic echoes.
This is where the scope of the problem transitions from public health to evolutionary biology.
It gets really deep here.
We're examining a 2025 longitudinal study published in JAMA Network Open, paired with groundbreaking 2026 research into the specific cellular mechanics of PFAS exposure.
And what do they show?
These studies confirm that our biological ledger doesn't zero out when we die. Biology records environmental experience in our physical structure, and it passes that record forward.
Let's clearly explain epigenetics for a second, because it's a concept that is fundamentally shifting how we understand human development.
It's vital to grasp.
Right. So if your DNA is the hardware, like the physical circuits you were born with, epigenetics is the software code that tells that hardware how to run.
Good analogy.
Your actual genetic sequence, the A, C, T, and Gs, doesn't change.
Yeah.
The environment can attach chemical tags to your DNA that dictate whether a specific gene is turned on, turned off, or dialed up.
Yes, and the primary physical mechanism for this is called DNA methylation.
Methylation.
It occurs when a methyl group, which is a carbon atom bound to 3 hydrogen atoms, physically attaches to the promoter region of a gene.
And what does that do?
When that methyl group attaches, it acts like a physical roadblock. It prevents transcription factors from binding to the DNA, effectively silencing or modifying the expression of that exact gene.
Okay, here's where it gets really interesting. Think of it like a massive soundboard in a recording studio.
Okay.
You have thousands of sliders controlling the volume of every single instrument. That soundboard is your genome. Epigenetics is the environment walking into the studio and pouring superglue over specific sliders.
I like that visual.
Some sliders get glued at maximum volume, some get glued at 0. The soundboard itself hasn't changed its structure, but the music it produces is entirely altered.
And it stays that way.
Right. And what the 2025 JMA study found is that the maternal diet is walking into the studio and gluing the sliders for the next generation.
Exactly. The JAMA Network Open study tracked maternal ultra-processed food consumption and correlated it directly with the child's developmental trajectory.
And what was the correlation?
They established a statistically significant association between high maternal UPF intake and an increased risk of early childhood obesity, as well as distinct alterations in neurodevelopmental markers.
Wow.
The highly engineered diet of the mother was actively modifying the metabolic set points of the developing fetus.
So before the child has ever taken a single bite of food, their internal baseline for how they process calories, how their insulin responds, and how their neurobiology regulates hunger has already been altered.
Altered by the hyperpalatable foods their mother was structurally forced to consume.
And the 2026 PFAS research isolates the chemical mechanism of this inheritance, doesn't it?
It does. It demonstrated that exposure to specific forever chemicals induces direct DNA methylation changes in both placental tissue and, crucially, in sperm.
Sperm and placental tissue, so it is coming from both the maternal and paternal lines.
Yes, both.
The chemicals migrating out of the grease-resistant wrappers are literally altering the methyl tags on the reproductive blueprints.
These are profound structural modifications to the fundamental regulation of the genes that build the human biological system.
It's hard to wrap your head around.
We are witnessing the environmental inputs of one generation, the disrupted leptin pathways, the circulating synthetic fluorocarbons, becoming the inherited biological baseline of the next generation.
It is exactly like handing down a corrupted software update.
That's exactly it.
I mean, it's one thing to run bad software on your own device, right? It runs hot, the battery drains fast, the sensors break. That's the metabolic override and the occupational hazard we
Of
discussed.
the individual cost.
But what this epigenetic data proves is that we aren't just damaging our own devices. We are effectively rewriting the default operating system for the next generation before they even power on.
Pre-installing the metabolic deficit at the factory?
Exactly.
The philosophical weight of this data requires a complete paradigm shift.
It really does.
The modern diet, characterized by persistent chemical exposure and ultra-processed intake, is no longer an isolated individual event. It is a state of forced continuity.
Forced continuity.
The boundary between a present individual exposure and a future generational legacy has totally collapsed. What is consumed today is permanently recorded in the biological architecture of tomorrow.
Which brings us to the ultimate synthesis of everything we've unpacked today.
Autonomy's last meal.
Yeah. We started by mapping the bloodstream inventory, detailing how carbon-fluorine bonds migrate from wrappers into our serum, remaining trapped for up to a decade.
The ledger.
We explored the metabolic override, explaining exactly how the physical destruction of the food matrix outpaces our digestion, while PFAS actively block our leptin receptors, blinding our brains to satiety.
Right.
We looked at the occupational reality, where the working class is treated like an industrial battery, pumped full of corrosive voltage to keep the supply chain moving.
The structural trap.
We dismantled the regulatory theater of the GRAS loophole, where the industry self-certifies its own safety while the FDA manages the visibility of the risk.
And the delays that causes.
And finally, we saw how this exact chemical and metabolic chaos is being methyl-tagged onto the DNA of the next generation. All of this forces a singular, unavoidable question about human autonomy.
It does. Because when you look at the entirety of these interconnected mechanisms, the concept of individual choice begins to disintegrate.
It falls apart entirely.
The system does not require you to make a catastrophic decision. It operates through continuity. It relies on the repetition of tiny, heavily constrained inputs.
Like a packaged meal during a long shift.
Or a drive-through run when time is scarce, compounded over years.
We treat every meal as a simple transaction. We are basically trading long-term biological stability for short-term energy, convenience, and time.
But the data proves the transaction is fraudulent.
Right. You aren't making a free choice if the very neurochemical sensors required to make that choice are being actively jammed by the environment.
That is the structural reality. If the speed and engineering of the food are designed to bypass your mechanical society triggers,
Yeah.
and if the persistent chemicals in the packaging are structurally altering the endocrine pathways that report fullness, then your internal data sensors are feeding you manipulated information.
Corrupted data.
You cannot make a rational, autonomous choice based on corrupted data. The transaction completes itself, the metabolic shift occurs, the chemicals accumulate, and the genetic code is tagged, all without requiring or even permitting your conscious awareness.
So what does this all mean for you? Next time you are exhausted, when you're pressed for time, and you grab that brightly packaged, highly convenient meal, how much of that is your active choice, and how much is just your altered biology responding exactly how the system designed it to?
As we process the sheer scale of this structural engineering, the most critical question may not just be how we regulate the packaging, but what the packaging is ultimately doing to us as a species.
That's the real question.
If our internal hunger cues are being systematically overridden by food architecture, and our genetic expression is being permanently tagged by synthetic carbon-fluorine bonds, at what point does human physiology fundamentally diverge from its evolutionary past? Are we adapting to the packaging, or is the packaging gradually, irreversibly becoming a part of us?
That is a thought that fundamentally changes how you look at the world, and it will be lingering with me every time I look at the grocery store shelves. Thank you for joining us on this deep dive. We encourage you to look closer at the mechanisms governing your everyday life, and to question the hidden transactions you make just to get through the day. Keep analyzing the structures around you. We'll see you next time.